when these arteries move ahead, it is branching into what we call as capillaries. Okay, so your tissues have a network of these capillaries. So as opposed to artery, these capillaries are what is highly permeable. Arteries are nearly impermeable. Okay, so when this blood flows to the capillary, the size of the capillary is around 10 micrometer. Okay, and the size of RBCs are 7 microns. Okay, so through these 10 micrometer tubes, these RBCs will flow. Okay, and along with that, your drug will also flow, like aspirin. Okay, so in the level of the tissue, at the level of the tissues, this aspirin will leak into the tissues from the capillaries. Okay, why? Because size of aspirin molecules is very small, less than 1 nanometer. So it can easily pass through the capillary into the tissue where the target lies, like whatever receptors or enzymes to which the drug binds. Okay, but liposomes are very big as compared to a drug molecule. It is 100 nanometer. It will not go into the tissue. It will not leak into the tissue. Okay, it will stay in the capillary. Then how we are trying to achieve targeting? By targeting, I told you the entire liposome should go into the target compartment. It should not go into the toxic compartment. So how do you achieve targeting? By using liposomes. So that is done using the biology of a particular disease. Like for example, you can see this is a fluorescent micrograph of a tumor tissue. Okay, it is different from a normal tissue, and how this is different from a normal tissue, and how do you exploit this difference in tissue for achieving targeting. So this difference in tissue structure between a cancer tissue and a normal tissue will make the difference in what targeting. Okay, like for example, I'll explain this. You can see this is fluorescent microscope, fluorescent micrograph. This green colored part is the cancer cells, and this red line, which you can see, is the endothelial cells. That is a capillary. Okay, so this red line is the capillary, and green colored tissues are the cancer tissues. Okay, now you can see this arrow. There are cells inside the capillaries. In normal tissues, cells will never grow inside the capillary. But since this is a cancer tissue, the entire this perimeter of the capillary is filled by cancer cells. Okay. So what is the difference between a normal tissue and a cancer tissue? There is growth inside the capillaries, and these capillaries become more and more permeable. Okay. So in normal tissues, the liposomes cannot leak because it is not permeable to the liposomes. But in this case, due to high permeability due to overgrowth of cells, the liposomes can also leak into the tissues, which in case of normal tissues will not happen. Okay, so this is the reason why the liposomes can accumulate in the cancer tissues and not in the normal tissues where the permeability is not normal. In this case, the permeability is highly abnormal, which leads to leakage of liposomes into the cancer tissues. So we exploit this property of cancer tissues to achieve targeting. So you can see this catchphrase is often used in cancer nanomedicine: enhanced permeability and retention. So I told you these cancer tissues are highly permeable, and the leakage of these tissues results in the accumulation of liposomes. Now what about retention? Now whatever thing is leaking into the tissues from the blood capillary is collected by the lymphatic vessels. Okay. Otherwise, what will happen? These tissues will get swollen because of leakage of excess plasma into the tissues. Okay. So this excess plasma which is leaking into the tissue is gathered by the lymphatic vessels. But in case of cancer tissues, what happens? This lymphatic vessels also gets blocked because of overgrowth of cells. Okay, so once the liposome leaks into the tissue, it will be retained in the tissue because it is not taken up by the lymphatic vessels. So that's why we call it as enhanced permeability and retention effect. So this is the theoretical basis of how a liposome works in cancer. In normal tissues, due to normal permeability, liposomes will not leak into the tissues, whereas in cancer tissues, because of enhanced permeability, The liposome will leak into the tissues and it will be trapped into the tissues because the lymphatic vessels are in working. Okay, so that you will get enhanced cytotoxicity in the target compartment, which is the cancer in this case. Okay. So this is a cancer in the skin of a rodent model of melanoma. Melanoma is an aggressive type of skin cancer. You can see this cancer tissues, and when you give a nanomedicine containing a dye, in this case it is a Evans blue dye. Evans blue dye is a blue colored dye. So you can see after injecting this Evans blue dye in a nano carrier, you can see after 6 hours, you can see accumulation of the dye in the cancer tissues. Okay, so this is classical EPR effect. You are giving nanoparticles, blue colored nanoparticles, dye containing nanoparticles, and the entire particles are going and depositing in the cancer tissues. Okay, so after 24 hours and 72 hours, it is retained. So not only that the drug is permeable, the liposomes or particles are permeable, it is retained for very long duration of time. Okay, this in practice will result in high cytotoxicity in the target compartment. So this is classically clear. Now this is the commercially available liposomes. It's called Doxil. This was approved way back in 1995. Okay, so this is now marketed by Johnson & Johnson. So this is the anatomy of the liposome. You can see that what is a liposome? Liposome is a vesicle. It is just like a, you can say, water balloon. Okay, so it has a membrane which is made up of phospholipids. Inside the membrane you have an aqueous compartment, and the drug will be in the aqueous compartment inside, and it is having a shell of phospholipids. Then above the phospholipids you see there is something called the PEG molecule. So inside you have the drug, and then you have a phospholipid membrane, and on top of that there is a PEG molecule. So why is there a PEG molecule? What is the function of a PEG molecule? See, whenever you inject some particles into blood, any particle, the immune system will recognize that particle as something foreign, and it will phagocytose that particle. Okay, so if the particle has to reach the target, it should not be taken up by these phagocytic cells. Okay, so we have to bypass these phagocytic cells so that the particle will reach the target. Okay, so for that we are coating the liposome with a PEG molecule so that the phagocytic cells are not able to recognize these particles. Okay, so this recognition system of phagocytic cells are blocked by this PEG molecule. So that is the function of PEG molecule. So whatever liposomes are injected into the blood will stay in the blood without being taken up by these phagocytic cells. Okay. Now coming to the regulatory part, there are three major regulatory pathways. Okay, 505(b)(1). Okay, so this is used by most of the new for new drugs which are coming into the market. The regulatory pathway is 505(b)(1). Second regulatory pathway is 505(b)(2). So these liposomal carrier nanoparticles are approved through a regulatory pathway which is called 505(b)(2). And what is that? How is it different from a new drug? It is an old drug you can see it is an old drug which is encapsulated in a new particle or liposomes. Okay, so this is different from a new drug. Therefore, the regulatory pathway also differs. So 505(b)(1) is for new drugs, 505(b)(2) is for modified drugs, and 505(j) is for generic drugs. You know what generic drugs are? Anyone? Generic. So off-patent molecules which can be manufactured by any industry or any company. Okay, other than the innovator company. Okay. So now there is a approval process called the accelerated approval process. Okay, what is the accelerated approval process? So this is the reason for the commercial success of liposomes, because they are able to get the approval process very fast, and that process is called accelerated approval process. Okay, So there are few conditions which are mandatory for getting accelerated approvals. What are those conditions? If a disease is serious or life-threatening. Okay. If a disease is very serious, like cancer, you will get accelerated approval for that. Second thing is when the drug appears to provide a clinical benefit not found in other drugs. Like if you are putting the drug in a liposome and if it is enhancing, if it is giving enhanced effect as compared to the pure drug injection, then it can be considered under accelerated approval. Third is when it is based on a surrogate endpoint. Now, what is a surrogate endpoint? Okay, so in case of in oncology, what is the endpoint of a clinical trial? Endpoint of a clinical trial means when is the clinical trial ending? What is the endpoint? Generally, survival is taken as the endpoint. Okay, so if you are giving an anti-cancer drug and if the patient is surviving for 5 years, then that will be the endpoint. If he survives for 10 years, then 10 years will be the endpoint. So this clinical trials can go endlessly if you don't fix a time period. Okay, so for some drugs it will be 2 years, 5 years, 10 years. Okay, so instead of the survival as the endpoint, you take an alternate endpoint which is called as a surrogate endpoint. And what is the surrogate endpoint? You will take the tumor regression. Tumor regression means after you have given the drug, what is the reduction in the tumor weight? You can see it by a MRI or a PET scan or a mammograph. Okay, after giving the drug, you can see that tissue is regressing. The cancer tissue is regressing. So that can be a surrogate endpoint instead of the usual endpoint, which is the survival time, which may be 2 years, 5 years, or 10 years. Okay. So this is a better endpoint because you can do it very fast. So these are the three conditions under which you get accelerated approval. Okay. Now this is all about the theoretical aspects of nanomedicine, why nanomedicine works and how it works and why it is better than a free drug injection because of its targeting capabilities. Now this is the next part of my talk wherein I am talking about the downside of nanoparticles. This is an article published in the Journal of Biomaterial, ACS Nano. So in this, the author states that several drugs has been tested against nanoparticles in clinics, right? Drug injection versus nanoparticles. Okay, and most of the nanoparticles have failed in this test. That means the anti-cancer effect of this drug injection is equivalent to that of the nanoparticles. It has no additional benefits. Okay, but in theory I told that it has additional benefits because of its enhanced permeability and retention. But in real practice, when you see the clinical results, there is no real benefit. And this paper highlights that thing. Okay. Now, what are the causes of this lack of actual benefit in case of nanoparticles? So this EPR effect has resulted in a lot of research paper, but in actual practice, liposomes aren't working that well. Okay, that is almost equivalent to the free drug injection, and I'll little bit spend some time on what is the reason for that. So EPR, I told you that EPR effect is due to the permeability of drug into the cancer tissues. Okay, so this EPR effect is not consistent. For some cancer, the EPR effect may be enhanced, but in some cancers like pancreatic cancer, there is a lot of extracellular matrix and EPR effect really don't work in those cancers. Okay, so EPR effect is limited to a certain type of cancer like breast cancer and all, in which the liposome may work. Okay, so the thing is EPR is not uniform in different cancers, and this EPR effect may change according to the stage of the cancer. Like stage 1, stage 2, stage 3 will have different EPR effects. Okay, so that is one of the reasons for the variable effect of liposomes. Another thing is, I told you the PEG is very immunocompatible and it prevents the phagocytosis of liposomes. but it is not really the case, because it has been found that those who take liposomes, pegylated liposomes, develop antibodies against PEG. Okay, and when antibodies are developed against PEG, these particles are again susceptible to phagocytosis. Okay, so in the first injection of liposomes, the antibodies are not developed, but when you inject the liposomes for the second time, these antibodies are developed and it will result in enhanced phagocytosis. This is reason number 2 for the lack of efficacy. And thirdly, the most important thing is these liposomes are tested in animal models. Okay, and animal models of cancer is not that great. You know how cancer is developed in animal models, rodent models. If you inject cancer cell in a rat, the rat won't develop cancer. Okay, you have to inject the cancer cells in a immunocompromised mice. Okay, which is called as the nude mice. So there is no immunity in that mice, and when you inject cancer cell, it will develop cancer. Okay, so we are testing the drug in an animal which is not having any immunity. So you are not able to study the effect of immunity on a nanoparticle carrier. And I told you that immune system is responsible for the clearance of these nanoparticles. Okay, so without a functional immune system, you cannot test these nanoparticles. Okay, so these preclinical models are not very good for testing liposomes. Now coming to the last part of my slide. So I told you that liposomes or nanoparticles will work in certain cases and will not work in other cases. Okay, so first you have to segregate the patient group as those patients who can get effective therapy out of liposomes, and for those patients who will not respond to liposomes. How can we categorize the patient into those two different categories? Patients who will get benefit and patients who won't get benefit. For that, you do a diagnostic. You prepare a diagnostic nanoparticles and then inject the patients with the diagnostic nanoparticles and check whether EPR effect is functional in those patients or not. Okay. Once you have identified that EPR is functional, then you give the actual nanoparticles. That is one of the way, and that will give you a personalized nanomedicine. Okay. So first we'll segregate the patients in which EPR is functional and the patients in which EPR is non-functional. That is done using a diagnostic nanoparticle. You will use a dye or an iron oxide nanoparticles and then get the PET imaging and will identify whether nanoparticles are accumulating in the cancer or not. Okay. Once you have identified that, then you will give the actual therapeutic nanoparticles. Okay. So that is one of the way. The future of nanoparticles is personalized nanomedicine. So this is the last slide. Thank you so much. The summary is that liposomes are effective, but effective only under certain conditions. In certain condition means in those patients who have a functional EPR. So once you identify the functional patients with functional EPR, you can give this therapeutic liposome. For other patients, this may not work well. Okay. So that is the bottom line of my presentation. Thank you so much for your patient hearing. If you have any queries, I will be happy to take it.
If you have any question, please ask.
Yes. Mine too.
Sir, you said that eastward code, they are increasing the country. So we use the finality of the product. But there is another product called metastasis. The metastasis, the tumor breaks in the final cells and should spread through the body, to the blood tissue and lymphatic tissue. So, is there any approach to deal with the metastasis?
See, this metastasis happens at a later stage in the cancer development. Okay. For those metastatic cases, most of the drugs are not working. Okay, if the patient has reached the metastatic state, it is almost not possible to recover. So liposome has no additional advantage in case of metastasis. This whatever I was talking about is are those cases in which there is no metastasis. In case of metastasis, none of the drugs are doing well. Okay, so that is an advanced stage of cancer. That is a very aggressive stage of cancer in which most of the drug fail. Okay, including this nanoparticle.
You should see that in case of anything, we are trying the VEGF, aspirin growth factor.
Yes.
So we can reduce the supply of the blood, so we can reduce the cancer.
Yeah, angiogenesis is another part. Okay, angiogenesis is the evolution of cancer cells. Okay, so for anti-angiogenic drugs are there, like Avastin is a antibody which is anti-VEGF antibody. Okay, so that is when you give an anti-VEGF drug, it will stop the growth of cancer tissues. Okay, the tissues will not grow bigger, but the existing tissue has to be removed. Okay, so this is used as an adjuvant therapy. So you will give chemotherapy along with this adjuvant therapy. So this adjuvant therapy will stop the growth of cancer cells, and this chemotherapy will destroy the existing tumors. Okay, so that is good, but it's not an alternative for like metastasis is a different case. For metastatic patients, it is very difficult to treat using normal chemotherapy. But one of the promising things now emerging is immunotherapy, which is highly promising because immunotherapy is good because you don't have this terrible side effects with associated with chemotherapy. Whenever you give drugs, chemotherapeutic drugs, the main problem is the huge side effect. But in case of immunotherapy, we can obviate or like we will have something which will not produce any side effects. Okay, like antibodies or use of any immunogens, cancer vaccines, etc. So that is a promising area now growing up. Thank you. Thank you. Yes. Okay. Thank you so much.
Thank you so much, sir, for letting us know about the importance of various secondary metabolites for the treatment of cancer. Thank you, sir. Now, I would like to invite Dr. Adityanath Pandey, sir, Professor and Principal of Pushpendra College of Pharmacy, Ambikapur, for summarizing your valuable knowledge about the session given by Dr. M.S. Sudhi, sir. Thank you, sir.
Firstly, I am very, very thankful to Dr. M.S. Sudhi sir, very acknowledgeable and fantastic and extremely, because I am also work in nanoparticles in cancers. Sir already discussed problems and possibilities on cancer, nanoparticles, nanomedicines. Sir already details in discuss barriers, epithelial barriers, endothelial barriers. And sir already discussed the right time and right dose and correction of durations. Sir already discussed in the liposomes, and conventional liposomes in markets are already available. Sir already discussed in brief blood distribution in the body, EPR effects, passive tumor targeting. So sir, again I have very extremely thanks. And I want to all students and participants, सर को सभी लोग थैंक्सफुल करिए, धन्यवाद दीजिए, क्योंकि सर ने बहुत ही शानदार सेशन लिया। और सबसे खास बात, ये जो सेशन आपने अभी लिया है नैनो टेक्नोलॉजी पे, आप लोग बी फॉर्म 6, 7 या 8 सेमेस्टर में आप इसको जरूर पढ़ेंगे। और आई थिंक जो भी बच्चे एम फॉर्म कर रहे होंगे या पीएचडी कर रहे होंगे, उनके लिए ये बहुत ही यूजफुल रहेगा। मैं चाहूँगा कि सभी लोग एक बार अपनी जगह पर खड़े होकर सर का जोरदार ताली से धन्यवाद। थैंक यू सर, थैंक यू, बहुत ही शानदार प्रेजेंटेशन था आपका। थैंक यू।
Thank you so much, sir. Life is self-representation, not a competition. Life is self-representation, not a competition. So all the participants, please take a step for the poster presentation.
I would like to request all the dignitaries to proceed for the poster visit in pharmacy department. Also, it's a time for tea break, so you can also have some refreshment. All the participants, please proceed for the poster presentation.
गाना गाऊंगी वहाँ पे प्रैक्टिस करूँगी अरे ये रख लो मुझे नहीं ये वो सब जमा देना कल से इसको लेने
हो गया मैम
छोड़ना है या क्या करना है क्या हाँ हो गया आज का
गाना
स्प्रे वगैरह कुछ होना है क्या करना है सब छुड़वा दो बच्चों को भिजवा दो तुम लोग लेके जाओ ट्रे ब्रेक का मैम जाओगे सब